بسم الله الرحمن الرحيم. دكتورة هدى مصلح محمود، كلية العلوم، قسم التقنيات الاحيائية. المحاضرة microbial genetic, second lecture. In this lecture I talk about the most often studied in molecular biology of prokaryotes is E. Coli. The genes of E. coli are located on a circular chromosome of about 4.6 million base pairs. This about 1.6 millimeter long molecule is compressed into a highly organized structure which fits inside the 1 to 2 micrometer cell in a format which can be still by the gene expression machinery. Bacterial DNA is supercoiled by DNA gyrase, chemical inhibition of gyrase without allowing the cells to reprogram gene expression relaxes supercoiling and expands the nucleoid, suggesting the supercoiling is one of the tools used to compress the genome. In this scheme, we show the coiling maintained by gyrase enzyme, relaxation of the coils by topoisomerase. Nucleosome formation. DNA is more highly organized in eukaryote cells. The DNA is associated with the proteins called histones eukaryotes. These are small basic protein rich in amino acid like lysine and or arginine. There are 5 histones in eukaryotes cells: H1, H2A, H2B, H3, and H4. This figure shows the nucleosome, which contain the DNA strand and the histone, H1 histone. This compound called nucleosome in eukaryotes. Based on stirring the DNA coils around the ellipsoid approximately about half and 1/3 or 1/66 base pair before proceeding to the next. The DNA plus the histone protein arranged in the formation are referred to as a nucleosome. The stretch of DNA between the bases varies in the length about 14 to 100 base pair. H1 appears to associate with the linker regions to enable the nucleosome to supercoil. When folding is the structure reaches maximum, the chromosome can be visualized. This figure shows the chromosome structure and the organism. Eukaryote replication. The nature of the DNA replication was illustrated by Nicholson and Stahl. This figure shows the parental helix and the replication fork and replex. Michelson and Stahl experiment grow the bacteria in heavy nitrogen N15 transferred as the first tube. The second tube only transfer the bacteria to N14 heavy nitrogen and then transfer the bacteria to the normal nitrogen N14 before bacteria reproduce in a new media. All bacteria contain the heavy DNA, such in the third tube and the fourth tube. The second generation contain 2 bands of the hybrid N14 and N15. Samples were taken 1 round of replication and 2 rounds of replication. This figure shows the semi-conservative replication of the DNA, which is the replication of the DNA in the organisms. Each original strand serves as template or pattern for the replication of the new strand. The new strand contains one original and newly synthesized strand. As this figure, the original strand and the duplicate of the strand are DNA. Eukaryote replication, multiple linear chromosomes. Each chromosome have some characteristics such as multiple linear chromosome. Each chromosome has more than one origin of replication, but the prokaryotes has one origin of replication. Approximately 1,040 times as long as bacterial DNA. So the eukaryote DNA or the eukaryote strand approximately 1,400 times as long as bacterial DNA. Multiple replicons on a chromosome because the multiple origin of replication. ORIS is along the length every 10 to 100 micrometer. Replication forks and bubbles are formed. Replication proceeds bidirectionally until the bubbles meet. Because the replication fork meets one to the other, and this is meet bubbles. This shortens the length of time necessary to replicate eukaryote chromosomes. This takes short time to replicate eukaryote chromosome because the chromosome of the eukaryotes more length than prokaryote chromosome. The process of elongation occurs at a speed of 15 to 50 to 100 beats per minute as compared to 750 to 1000 beats per minute. The origin of replication. This figure shows the origin of replication and replication fork. The replication and the origin of replication bidirectionally, about 10 to 100 micrometer, the space between two replication fork. Eukaryote replication. During the S phase, there are 100 replication complexes, and each one contains as many as 300 replication forks. These replication complexes are stationary. The DNA threads through these complexes as single strand and emerges a double strand. DNA polymerases. 14 DNA polymerases have been observed in human beings as compared to 3 in E. Coli, such as polymerase 1, polymerase 2, and polymerase 3. So, there are differences between DNA polymerase in eukaryotes and prokaryotes. About 14 DNA polymerases enzymes have been observed in human beings as compared with E. coli or prokaryotes, which contain 3 types of polymerase. Prokaryote replication. This table shows the different enzymes which play a role in the replication, in the formation of the replication fork. Bacterial topoisomerase, examples such as bacterial topoisomerase 1, vaccinia virus topoisomerase 1, eukaryotic topoisomerase 1, reverse gyrase topoisomerase 5, DNA gyrase topoisomerase 6, T4 topoisomerase 2. The source of these enzymes is the first type E. coli, vaccinia virus, calf thymus, thermophilic bacteria, hyperthermophilic bacteria, E. coli, and bacteriophage T4. These enzymes, each of these enzymes play a role in the replication fork, such as the bacterial topoisomerase 1 play a role in the relaxation of the negative but not positive supercoils, while vaccinivirus topoisomerase 1 play a role in the relaxation of positive and negative supercoils. So the type 1 is work on the negative supercoils but not positive supercoils, while the vaccinivirus topoisomerase 1 work on relaxation of positive and negative supercoils. The eukaryotic topoisomerase from the calf thymus play a role in the relaxation of positive and negative supercoils. Reverse gyrase from the thermophilic bacterium introduces positive supercoils into DNA. Topoisomerase 5 hyperthermophilic bacteria relaxation of positive supercoils. DNA gyrase introduces a negative supercoils into DNA, while topoisomerase 4 DNA relaxation and potent dictination. Topoisomerase from the T4 bacteriophage T4 relaxation of positive and negative supercoils. Bidirectional replication, there is an origin of replication such in this figure. Step 1, the origin of replication of the double strand. 2, replication forks are formed in two directions. Replication occurs around the circle until they have opened and copied the entire chromosome. So the replication occurs until the two replication fork meet entire the chromosome and copied in the same time. Replicon contains an origin and is replicated as a unit. ORI, origin of replication. Characteristics used to define origin: the position on the DNA at which replication start points are found. A DNA sequence that, when added to a non-replicated DNA, causes it to replicate. DNA sequence whose mutation abolishes replication. A DNA sequence that in vitro is the binding target for the enzyme. This is the characteristic used to define the origin of replication, ORI. Different types of topoisomerases. The topoisomerases, when the double helix of DNA, which is composed of two strands, separates, helices make these two strands rotate around each other. The DNA protein is the helices most involved in replication, but the protein may also participate in unwinding. The single-stranded binding proteins SSBB help to keep the strand open. But there is a problem due to the topological reasons that the unreplicated part ahead of the replication fork will rotate around its helical axis when the two strands separate at the replication fork. Topoisomerase action, it causes strong strain in the helix 1. Thus, it is impossible to unlink the double helical structure DNA without disrupting the continuity of the strands. In order to perform an unraveling of the compensing winding up, DNA enzymes are required. Topoisomerase change the linking number as well as catalyze the interconversion of the kinds of topological isomerase of DNA. Initiation. The initiation ORIC, origin of chromosomal, means the origin of chromosomal replication, recognized by DNA. A protein only recognized if the sequence GATC sites are fully methylated. So, after the methylation of the sequence GATC, the origin of replication recognized by DNA protein. This is the first step. Binding of the DNA protein allows DNAB to open complex. DNAB is the replication helicase, means the enzyme replication helicase, which unwinding the DNA strand separation by helicase, and the single strand binding protein keep the strand apart. DNA gyrase topoisomerase puts swivel in the DNA, which allows strand to rotate and relieve strain of unwinding. Explanation. Recall the DNA double helix is tightly one structure and that the bases lie between the two backbones. If these bases are the template for a new strand, how do the appropriate enzymes reach these bases? By the unwinding the double helix. The enzyme called helicase catalyzes the unwinding of short DNA segments, which is called the helicase, just ahead of the replication fork. The reaction is driven by the hydrolysis of the ATP, adenosine triphosphate. As soon as duplex is unwound, the single stranding binding protein binds to each of the separated strands to prevent them from the base pairing again. So, the single strand binding protein keep the stranding to unpairing or open strands and prevent them from the base pairing again. Therefore, the bases are exposed to the replication system. The unwinding of the double helix or duplex would cause the entire DNA molecule to swivel except for the action of a topoisomerase DNA gyrase, which introduces breaks in the DNA just ahead of the unwinding duplex. These breaks are then rejoined and after a few revolutions of the duplex. The need of a primer. When the DNA template is exposed, DNA synthesis must begin, but DNA polymerases not only need a template but also need a primer for the replication to proceed. After the observations, the RNA synthesis is required for DNA synthesis. It was discovered that the synthesis of DNA fragments requires a short length of RNA, which is defined as a primer. Primosome means the complex of 20 polypeptides makes RNA primer in E. Coli. A primosome contains a primase. Primosome moves along DNA duplex in the direction 3' to 5' with respect to lagging strand, follows the replication fork. Even though primer is made in the direction 5 to 3', note the symbol means indicate that the direction that is the primer is made from the 5' to 3', and protein removes single N' protein removes single-stranded protein in front of primosome, which is binding the strands from the winding again. DNA A, B protein reorganizes some components of primosome and prepares DNA for primase. Primase forms the primer. This figure shows the double helix or DNA double helix, the 5' end and the 3' end, and the other strand from the 3' end to the 5' end. Complex that synthesize most of the DNA copy continues the DNA polymerase enzyme and other proteins. The gamma delta complex and the B subunit of the holo enzyme bind it to the template and the primer. The alpha subunit carries out the actual polymerization reaction. Holo enzyme, all the proteins form a huge complex called Replication. DNA polymerases 3. This is a stationary complex that probably attached to the plasma membrane. The DNA moves through the replication and is copied. This figure shows also the double helix and the ground end nucleotide ID. Elongation of the chain need the deoxycytosine triphosphate and deoxycytosine monophosphate, which means deoxycytosine monophosphate plus the pyrophosphate. Energy is supplied for the biosynthesis by the cleaving of the phosphate bond. And this figure shows the DNA polymerase reaction, which means the 4 type of a nucleotide, deoxyadenosine triphosphate, deoxyguanosine triphosphate, deoxycytosine triphosphate, and deoxythymine triphosphate. By DNA polymerases, which result as DNA plus pyrophosphate. And this mechanism shows also the mechanism of the chain growth, which contain the 4 types of the nucleotide, also the sugar and the phosphate group. Elongation proceeds in 5' to 3' direction and requires number 1, all 4 deoxyribonucleoside, 5' triphosphate, deoxyadenosine triphosphate also, and so on. Magnesium ions, a primer made of the nucleic acid, and the DNA template. Rate of elongation about 750 2000 nucleotide per second. Rate of formation of initiation complex equal to 1 to 2 minutes. Elongation DNA polymerase 1, type 1, type 2, and type 3 in E. Coli. DNA polymerases type 3 holoenzyme complex of 7 polypeptides. Replication, primosome, and 2 DNA polymerases type 3 synthesize DNA on both strands simultaneously without desiccating from DNA. DNA polymerase type 3 catalyze the addition of deoxyribonucleotide unit to the end of the DNA strand with the release of inorganic pyrophosphate PPI. DNA residues plus deoxynucleotide triphosphate means DNA plus residue plus pyrophosphate. Attachment of a new unit is by their phosphate group to a 3-hydroxyl and of a resisting DNA chain. This figure shows the replication fork and the direction of the replication fork, the leading strand and the lagging strand. And this figure also shows the direction of the leading strand from the 5' to 3', and the direction of the lagging strand from the 3 to 5. Also show the Okazaki fragment and the primer, RNA primer. Also the next in the lagging strand, single binding proteins, single strand binding proteins, also the DNA gyrase and the helicases. All these enzymes work equally or together on the double helix or the DNA strands to start the formation of the replication fork by DNA gyrase, DNA helicases, and single strand binding proteins, also the different topoisomerases enzymes, and to form the leading strands also in the other hand the lagging strands and the Okazaki fragment. This figure shows the synthesis of the leading strands and the extension of the strand, lagging strand and leading strands, and the replication growth, most recently synthesized DNA, and the growth of the Okazaki fragment in the lagging strand. The lagging strand and the discontinuous replication, the replication on the 5' to the 3' strand differs. The template strand will still must be read from 3 to 5'. The reading begins at the replication fork, occurs the same time as the synthesis of the lagging strand. Okazaki fragment of the lagging strand has been duplicated by the formation of the Okazaki fragment. DNA polymerases 1 or RNase H removes the RNA primer. Polymerase 1 synthesizes the complementary DNA to fill the gap resulting from the RNA deletion. The polymerases removes one nucleotide at the end. Ligase can catalyze the formation of the phosphodiester bond given and attached by the adjacent by binding the adjacent 3OH or 3' OH and the 5' phosphate. This can fill in the unattached gap left when the RNA primer is removed and filled in. The DNA polymerase can recognize the bond on the 5' and the other 3OH in the other hand. At the end of this lecture, we explain the replication fork and the steps and the components of the replication fork or the replication of the DNA bidirectionally in the eukaryotes or prokaryotes.